Warren will be presenting on nature-based solutions addressing biosalts management and treatment efficiencies. So if we can please welcome her to the stage.
So I first want to say thank you so much for coming out. I was saying when I landed today, I saw blue sky as I was landing, and I came out of the airport and saw this. So it's great to be here, and I'm so appreciative that you guys all turned out. And I'm going to talk to you about sulfur, and I'm going to talk to you about bugs. And that was a great introduction. Thank you. It's always weird to hear people talk about your pioneering work. Is it though? Is it? But we are going to talk about microbes and why it's interesting to think about that. Nope, that's not mine. Yep, so I just want to give a shout out to CIM. Being part of the Distinguished Lecturer Series is really fantastic, and it's a way to come and talk about research and what we're doing. And they fund this, which is awesome. I suspect most people in the room know this, but over 70% of the world's metals come from sulfide-hosted minerals. And so as the world is looking to scale up to support the energy transition, think about copper in particular, we are going to see more sulfide mineral processing. The challenge with that, I only have to say acid rock drainage. I think everyone in the room nods their head. It remains the global #1 pollution issue for the mining industry, despite having been discovered and researched for over 100 years. So we know that this is an issue. And negative impacts, of course, for the mining industry. There are so many legacy cases around ARD. It does not help when you're trying to scale up. And so there's this real concern, this tension between how do we scale up to support the energy transition, whilst also making sure that we do not catastrophically destroy already stressed freshwater resources. We've got to figure this out. And so one of the ways we can think about this, and as is being done in many other sectors, is we start thinking about, are there opportunities for nature-based solutions? And I suspect this is something a lot of people in the room are thinking about, heard about. I think about nature-based solutions as a way, are there biological processes happening in your systems? If so, when we characterize them, can you do it more efficiently? Can you do it more sustainably? It's not necessarily that we're going to increase biodiversity or have better outcomes for habitat, which obviously we want, but we're thinking about harnessing those endemic bugs that are in your system to do something for you, or to at least shut them down if they're doing something you don't. And so this idea around sulfur has been known for a long time, but we really have not done a very good job of characterizing the bugs that are involved. And so, as I mentioned, ARD is sort of the poster child for microbial processes that have been known about for a long time. We still don't have source control under control, right? So you go to most mines, they still are not sure when they're going to get ARD, if they're going to get it, and when it does happen, how do you turn it off? We know at its heart, the recipe for ARD, you need some sulfides, you need some bugs, and if you've got water and oxygen, you're going to get ARD, more than likely. But beyond that, the process design of that, the controls, the levers, are still not well understood. And partly that's because it's complicated, and partly because the technologies have taken a while to develop to a point where we can use them effectively, especially for the bugs. So we work with quite a few base metal mines across the country, and we were called in to work on this problem, thiosalts, which for the industry, and I came in as a geochemist, biogeochemist. I didn't know what a thiosalt was until the mining guys told me, oh yeah, it's the sulfur stuff that comes out of the mill. And I'm like, oh, okay. And it's an industry term, right? For these intermediate sulfur compounds that come out in the tailings, end up in your tailings impoundment. And for 30 years, the Biosalts Consortium in Ontario have been really trying to manage that issue. Because part of the issue with thiosalts is that they're not all destroyed by current treatment. And so some of them were making it through treatment into the receiving environment. The second part of the problem was that mines did not have accurate, robust monitoring practices in place. So if you can't monitor it very well, your treatment doesn't remove it all, and then all of a sudden you have a problem in your receiver and you don't know why, right? So you can see how that problem was kind of pushing things. And so when we were called in, it was to start thinking about, okay, we've been looking at this from a very much an engineering point of view. And trust me, being in an engineering department, I love because it's practical. We need to come up with solutions. But sometimes you need to back up a little bit and think about what's really going on at the systems level. And is there a way for us to come up with a better solution? So when we were called in, the companies were struggling with the methods that they had. They were sending samples off site. They'd have to wait 3 or 4 weeks to get that data back. So it's very hard to intervene if you're waiting a month to even know if you've got a problem. But they also knew that the treatment they had was not effectively capturing all of those potential sulfur compounds. And so the risk is if it makes it offsite to your receiver, you start having acid rock drainage, you're in non-compliance, you start to see those environmental issues ramping up. And so when we came in, one of the reasons, and I'm really proud about this project because it was an industry pull project, which is why I think it was so effective, because industry was all in terms of partnering with us and really opened the door to saying, yeah, we're going to let you do something different because we need a different solution. And so when I came in to talk to them about what we were thinking of doing and looking at what was going on in tailings impoundments and in receivers, one of the things I said was what we want to do is we want to look at the chemistry, absolutely, but we want to look at the bugs as well. And that was a little bit of a quiet moment in the room. But then as we started to talk to particularly the engineers and the water engineers, it was like, look, your models are failing. They're not predicting what's happening. You know your treatment isn't working. There's something that you're missing. And I'm pretty sure it's the bugs. So let us play with that. And one of the things I will say is, if you take nothing from this lecture other than this point, mines are biologically reactive systems, especially your waste, your ore. You're bringing up reactive compounds, you're exposing them to water and air, you're creating McDonald's for bacteria. They will be in there. The question is, who's there? What are they doing? How fast are they doing it? Is it good or bad? Very simple questions that we now have the tools to answer. One of the reasons we struggle is that mining environments are particularly poorly characterized, so we don't know the endemics in your systems. We can extrapolate from other systems, but we don't have a good database yet across mines that start to build robust models. So if you're thinking about it, do it. It will really help your best lines. And so we started with this premise, and I just want to say, you'll hear me say some of these words. Any of it, feel free to ask me a question. Microbiome, I suspect, is this something everyone's familiar with if I say that, right? We talk about human gut all the time, the bugs that live in your system and essentially are now linked to all kinds of disease, mental health. They essentially are the living community that control the processes that happen. We now know that those specific communities happen everywhere. So if we were to take a swab off one of these chairs, or we went downstairs and took one from outside, there'd be a specific group of microbes that are living in each of those contexts. The point is that they're invisible engineers. They're working in those environments, they're interactive with it. And so when we start thinking about whether I'm thinking about soils, and the advances that we've made now that we've got these genomics technologies, right? Molecular techniques, extract the DNA. We can look at questions about identity, questions of function. How are those microbes making a living? What metabolisms are they carrying out? Think about it as your process engineers. Which pathways am I activating, right, in a reactor system? But we also can use the genes, right, the enzymes. We can look at which ones are turned on as a very accurate indicator of which reactions are firing up. And this is being used in all kinds of things. Mining has been slow to the game. I know we're fast followers, love to be second, blah, 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 right? But we need to get on board. And so the technologies have increased tremendously in power and have come down in price tremendously. What took the human genome about 10 years and about $3 billion, you can now do in about 2 days for a couple of $100, right? So the power and the cost of it has come to a point where we can use it effectively. So microbiome science, we're asking questions. Think about, please, whenever you're back on your mine site, look at anything, your ore, your waste, think about it interacting with water, know that you're going to have invisible microbes that are interacting with those materials. We can ask those same questions. Who are they? What are they doing? How are they doing it? And it will inform our understanding of system level outcomes. And it identifies essentially new levers that we can use. And so in systems where, usually I'm called in because mines are starting to see problems and they can't explain why, right? Which is never a good situation. You're reactive, you're scrambling to fix something, but you're not sure why it went wrong. But it usually tells you that you're missing a piece of the puzzle. And oftentimes in these waste contexts, because they are so biogeochemically reactive, there will be microbes involved. And that's really been a big piece of what we've been trying to untangle. I will say, because I've been asked about this at a few of these lectures, nature-based solutions, you can look it up. There are many, many definitions. I particularly like this one because it's very practical, right? So they're inspired and supported by nature, cost-effective, right? If nothing else in mining, we like to be practical. It's got to be effective, but it also will simultaneously provide environmental, social, and economic benefit. And so thinking about what's good for the environment ultimately is good for us, but it will also be good for your bottom line. And so we started to think about nature-based solutions was not a term that was being kicked around in 2016 when we started talking about this project. But this is exactly what we were doing. It was a case study to examine, could we do this kind of investigation where we're looking at these invisible engineers and come up with better solutions? That's a nature-based solution. So, and as I said, for thiosalts, particularly with our mining partners, they lacked the tools. It wasn't that they did not want to be successful. They lacked the tools to do it, which opened the door for them to think a little bit differently and allow us to do something which was a little bit out there in terms of thinking about mining systems as biogeochemically reactive, that we were going to consider microbiology. So this case study was really a push-pull from industry. So Hudbay is a huge partner on this project. 777 and the Anderson mine are big parts of the case study, as was Sudbury INO. And then we had Rambler, which is a smaller mine out in Newfoundland. And so we had a range of base metal mines, all processing different ores at different ages and stages, slightly different systems, but they all bought in. We had a multidisciplinary team, and it was a pretty big project that we were working on. As I said, we went across the country, and it was, I think it was the global first project that did anything like this. We are still the only project in the world that's done anything like this. And so, opening the door based on a Canadian study to what needs to be done globally, we're starting to see sort of the uptake. And really, particularly Shirley Milt, for those of you guys who know Shirley from Hudbay, was a huge player in getting this project on the books. So very straightforward questions. And the first one we wanted to look at, because systems-based, integrating biology is not rubber stamping, just collecting bugs. It's trying to understand your system better so that you can manage it better and have better outcomes. And the first thing we really wanted to tackle was, what are mines using right now to measure these thiosalts? Are they adequate? And is there a better method that we could come up with? And then we went into looking at, okay, do we have sulfur oxidizing bacteria, the guys that drive acid rock drainage in the tailings impoundments? People tend to think about waste rock for ARD. You don't tend to think about sulfur cycling in tailings impoundments. So we wanted to look at that, and we wanted to look to receiving environments. But we'll start with the methods first. Little bit of chemistry, and really just to point out that. And as I say, this is where language is so important, having partners willing to talk to you, because I needed to understand from a mining perspective, what the heck are these thiosalts? What do they mean by this? And what they meant were two particular sulfur species, thiosulfate and tetrathionate. And the reason that they consider those to be the dominant forms is because that's what comes out after processing of a sulfide mineral ore in the tailings. And because the perception is that your tailings impoundment is basically static, if that's what's coming out in your tailings stream, we model and manage that. Bob's your uncle, you're good to go, right? Not considering that the tailings impoundment is no different than a lake or an ocean. You're going to have biological cycling. And sulfur in particular is jet fuel to a lot of bugs. They like to play with it. And so we wanted to examine this idea. Were they the most important? If not, was there a better way to manage and predict what that sulfur risk might be? Because anything that's not sulfate, any sulfur compound, it's a thiosulfate, tetrathionate, sulfite, anything less than sulfur 6+ is capable of oxidation, which means it can generate acidity, right? All of those problems with ARD. And so the method we came up with is straightforward, mass balance, right? And so any of you guys who are doing this, you have this in your monitoring data. You will get total sulfur and sulfate. You subtract sulfate from your total sulfur. The amount of sulfur atoms that are left are all reactive. It's agnostic of what species you have. You still need to do speciation if you're interested in that, but it's saying you have this amount of reactive sulfur in your system. So it's highly conservative of risk for oxidation of that material that could lead to ARD. And so we conceptualized, let's start with a mass balance. Total sulfur minus sulfate. What's left, we're going to call it reactive sulfur. And what we did was we looked at that, but then we also looked at these two particular measurements that our mining partners were using, and we sent samples out to the external labs that they were using. So we could look at those measurements from the QA/QC labs for thiosalts or bulk thiosalts, and then we compared it to reactive sulfur. And any bets on what we might have seen? This is what we saw. And I'm only showing you a few of the data. We have over 60 data points if you want to look at the paper. But any of the blue points, which are higher, are reactive sulfur. And then the green and the orange are those thiosalt measurements that the mines were using for their monitoring purposes. The line kind of shows you that we're also picking up that reactive sulfur offsite in the receiver. So this kind of confirmed that not only was sulfur making it out, but a lot of it was in forms that are not thiosulfate or tetrathionate. It's something else. And so we knew that there was this relatively large pool of sulfur that was not being captured by current monitoring processes. What was really great was that Hudbay used our data to take this to the regulator, and they got approval within 2 months to start using this new method. They're the first mine and the only mine in the world right now that is using this to report on sulfur in their wastewaters. And so the Saskatchewan regulator was really happy. They could see the evidence. It's more conservative of risk. It's actually cheaper. You get your data already from your monitoring data, and it's directly comparable across mines. Because that was the other thing we found out, was that different labs are using different methods, but reporting them as thiosalts. So then you're trying to compare across mines and it's a different method. So this, it's published, very easy to do, highly recommended. And talk to Hudbay if you want any advice on how they implemented it. But what this also told me, going back to what comes out of the tailings, mostly thiosulfate and tetrathionate, that's not what we're seeing in the impoundments or the receivers. So something's happened to that sulfur, right? It's changed, which is also usually a signal that you've got biological processing happening. And so, when we look at sulfur in terms of an abiotic model, which is what all mines are pretty much using, any reduced sulfur in the presence of oxygen is going to oxidize, you're going to create sulfate and acidity. And for the mines, they're looking only at these thiosalt species that are essentially generated in their tailings and deposited in their impoundments. And of course, you deposit them underwater because you're limiting oxygen exposure, which is the main driver of ARD. So when we add bugs to the mix, and for thinking about sulfur, why might it be so interesting to bugs? First of all, there's a whole range of oxidation states of sulfur, multiple species. It means that bugs can catalyze many reactions to get energy. Sulfur is probably the oldest metabolism on Earth, right? Present way before we had oxygen. And so while we think of mines as being harsh environments, they are McDonald's for lots of bugs. They love them, right? Lots of energy to get. But it makes it more challenging for us. There's a great deal of flexibility and sort of dynamic changes in the way they process sulfur. They don't always take it all the way up to sulfate. They can do all kinds of recycling. Makes it a little bit more complicated. Doesn't mean we can't figure it out though. That's the thing to remember. And so we wanted to sort of start investigating what was now kind of demonstrating from the data, there was more than likely sulfur cycling happening in these systems. And so we wanted to look at, first of all, we know that sulfur oxidizing bacteria occur. Anyone been to Yellowstone? You go to the hot springs and lots of sulfur oxidizing bugs there, right? Because you've got that reduced sulfur coming up. It creates the energy for these bugs to use it. Same in the oceans. You see sulfur bugs processing these kinds of compounds. But in tailings impoundments, this was the first study that was really doing this cross-line assessment. And we wanted to see, were they different than the sulfur oxidizing bugs we might see in the ocean or in hot springs? Was there a distinct microbiome that was specific to these mines? Because if there is, then that means we have a biological agent we could use across mines, right? And that was really one of the goals of this. It's not just to identify the bugs. We want to use them as nature-based solutions. So to do this, anyone who's interested in this kind of work, and I know there are people in the room who are already doing this kind of work, which is awesome. You know, when you think about any water system, think about it as a biologically integrated system. You need to capture seasonal and spatial variability. So first thing is, if you're only ever collecting water at the side of your tailings impoundment, pretty good bet it's not going to capture what's happening 40 meters down in the middle, right? And so thinking about that spatial importance to zonation of process is really important. But then we integrate chemistry analysis with the bugs. And I'll talk a little bit more about that. So we characterize what's going on in the field, but then we bring back samples and we play with them in the lab, right? So we can look at not just which bugs are there, but what are they doing? What reactions are they driving? And what are the controls? And this is all to build a biologically integrated systems understanding. And I'll talk about that in a minute. So the first thing I want to point out, so this is just a very simple pie diagram, 16S data, which are basically, it's the cheapest data you can get. It basically gives you labels, right? It gives you names of different bugs. So if we get 16S data, you'll hear that, or DNA data. It's the cheapest, it's the easiest, it'll give you genes of identity. What came out of that was across all of our mines, and it was over 400 samples that we analyzed. So across mines and times, we're looking at a lot of them. There was a consistent set of sulfur oxidizing bacteria that were present in all those mines, which is great, right? If we're trying to develop a biological model, I need to see similarities across all those mines. But what was also interesting was that we saw the same sulfur oxidizing bugs in the receiver. That connection between the mining wastewaters and the impoundments and then in their receivers was there. Over 40% of these bugs are novel. They are not present in any other databases, i.e. other systems. They don't look the same as what we're seeing in these mining systems. Again, very, very clear signals of bugs that are well adapted to these kinds of contexts. But we were interested, does it matter that I have these different ones? Does it matter which SOB I have, or just are they all doing the same thing? And so we really wanted to look at then, does the label matter? Are they doing different things under different controls, which would help us build the model? It's the end of term and I taught 2 courses this term, so my voice starts to give out. What we saw looking across all of the mines, this is just showing you the percent abundance. And so when we look at microbes, we often look at functional groups. So in this case, I'm interested in the ones that are eating sulfur, right? And so I'm looking at how many are there in those communities. Normally, if you've got very high abundance, you know that that's probably an important pathway that they're processing. But you kind of see there's a lot of green across all of them. There's variability in how much average abundance across the mines. And then in our older mines, 777 and Sudbury Middle, we see some of these yellow and red guys popping up. And so I'm going to come into that in a minute. But one of the things I will say for anyone who's thinking about this or struggling with how do I interpret microbial data? Where is it useful? How do I apply it? 16S is very useful for giving you names. But beyond that, you're going to have to do a bit more work to know what are they doing and are they actually doing it. But it's a good start. And so what we saw was that when we started to look across our mines in terms of the green guys and the yellow guys, and I'll come to why we sort of separated them in a bit, was that they were definitely partitioning as a function of pH of the system, but also that reactive sulfur concentration. So we started to see that essentially there were ecological or essentially chemical drivers that were partitioning which of these bugs were present. And that to me tells me that they're doing something different, or at least different controls are going to activate some of these bugs differently. And so we wanted to then try to examine, and again, we had nothing to go on because in the natural systems, people often characterize sulfur oxidizing bacteria, but they're not thinking about levers and controls the way we do in a mining system, where I want to know what are they doing, does it matter, what controls it? In a natural system, it's more, we had some sulfur oxidizers, they're this kind. So we didn't have anything to template our mining systems against, but we knew that we needed to understand, are they doing the same thing? Does it matter if they come from 777 or from Sudbury INO or from the summer or the winter? Does it matter? Does it matter if they have a different name? And so we can grow them in the lab. And so we take samples from all of our mines. We had over 300 now that we've grown under the same conditions. And then we look at what they do with sulfur. And probably you can't see it just looking at these flasks. But if I said to you, I had 300 samples, I gave them all the identical meal, did they generate the same amount of acidity and sulfate? The answer is no, they didn't, right? So we know it matters which ones you have. And what we saw, and so what you've got here, I don't have a pointer, but basically the bars represent the amount of sulfate to acid that was generated in these experiments. These are for different parent waters. It could be a tailings impoundment or a receiver, different mine, doesn't matter. And then what I'm showing you is the abundance of the green or the yellow sulfur oxidizing bacteria. And what you can kind of see is that the ones that generated the most acid to sulfate, i.e. your fast acid rock drainage process engineers, have lots more of the yellow guys, right? The green guys are not producing as much. And so we started to call these guys fast and slow. And so we knew that it became very important to know which sulfur oxidizers you had, because which reactions they were catalyzing obviously was different with different outcomes. And so this started to set up our model in terms of actually it does matter. And so now we knew this, that we started to see that there were differences. We wanted to understand why there were differences, but then also what controlled that, because that's how you start to build a management strategy to manage what's happening in your tailings impoundment. I think what's also important to know, because lab work is always beautiful on the bench, right? The trick is, does it map out when you go back to field? And when we looked at some of our receivers, particularly for Hud Bay properties, where they had a long-standing acid rock drainage issue in Flin Flon, you can see we saw the similar trend, that where we had receiving environment samples that were low pH, we had much more of the yellow-orange guys, right? So